Hello and welcome to SAM Biology class. Friends, today we shall discuss about an enzyme called FoF1 complex or ATP synthase. This is also known as FoF1 ATPase. The transmembrane electrochemical proton gradient generated by the redox systems of the respiratory chain in mitochondria and in aerobic bacteria is utilized by proton translocating enzyme known as ATP synthase to catalyze the synthesis of ATP from ADP and inorganic phosphate. Similar enzyme also operates in chloroplast. This enzyme consists of two principal components, both of which are multimeric proteins. The components are FO and F1. FO complex contains three types of integral membrane proteins such as A, B and C. In yeast mitochondria and in bacteria, the most common subunit composition of FO complex is A1, B2 and C10. That means in yeast mitochondria and in bacteria, FO complex consists of 1 S subunit, 2 B subunits and 10 C subunits. Whereas in animal mitochondria, FO complex contains 12 C subunits and those in chloroplast contains 14. So these are the subunit compositions of FO complex in various organisms. In all cases, the C subunits are arranged in a specific manner to form a ring in the plane of the membrane. The other two subunits, such as A and B, are rigidly linked to one another but not to the ring of C subunits. So, C subunit, which are arranged to form the ring, A subunit and B subunit together constitute the FO complex. The other component of the enzyme is F1. The F1 component is a water-soluble complex of 5 distinct polypeptides such as alpha, beta, gamma, delta and epsilon. Each F1 complex contains 3 alpha and 3 beta subunits and 1 subunit of gamma, delta and epsilon each. The lower part of the F1 gamma subunit is a coiled coil that fit into the center of the ring of C subunits. The F1 epsilon subunit is rigidly attached to the gamma subunit and it also makes rigid contacts with several C subunits of the ring. The F1 alpha and beta subunits are arranged in alternating order to form a hexamer that rests atop the single long gamma subunits. So 3 alpha and 3 beta subunits are arranged in alternating order to form a hexamer which rests on the single long gamma subunit. And next, F1 delta subunit is permanently linked with one of the F1 alpha subunit and to the long 2B subunit of FO. So in this structure, FO, A and B subunits and the F1 delta, alpha and beta subunits together form a rigid structure that remain anchored in the membrane. The long 2B subunit of FO act as a starter that prevent the hexamer from moving while it rests atop the single long gamma subunit. When ATP synthase is embedded in the membrane, F1 particles protrude as a knob-like structure from the cytosolic phase. Experiments have demonstrated that some mitochondrial vesicles from which F1 particles are removed cannot synthesize ATP, but when F1 complexes are reassociated, ATP synthesis takes place. Because F1 particles removed from the membrane can hydrolyze ATP, they are also called as F1 ATPase. However, their role in the cell is to synthesize ATP from ADP and inorganic phosphate. The FoA subunit contains two proton half channels. Half channel 1 allow protons to move one at a time and to bind to aspartate 61 at the center of one of the C subunit lying at the middle of the membrane. Whereas half channel 2 after rotation of the C ring allow the proton to dissociate from aspartate and move into the cytosolic medium. During flow of protons, the C ring and the associated F1, gamma and epsilon subunits rotate as a unit. This causes conformational changes in the beta subunit of the hexamer, which leads to the synthesis of ATP from ADP and inorganic phosphate. So friends, this is all about FoF1 ATPase and its mechanism of action. For more updates, subscribe my YouTube channel, Sam Biology Class.